So I'm Ramesh Pillai. I was group leader at the Grenoble site starting 2006 till 2016. Before that, I was in Switzerland. I was doing my postdoc at the Friedrich Miescher Institute, FMI, in Basel.
One thing that I'm always asking is how did you get to know of the opportunity of working at EMBL relative to other places that you could go and set up shop?
Yeah, I think that's a very interesting story because when I was finishing my postdoc, I was talking to my PI there, Vitek Filipovic, and he was saying, okay, there are these institutions that you should try, and EMBL was one of them. And strangely enough, the week after we had that initial chat about what are the next steps, Ian Mathai was visiting FMI. So then at that time I had the opportunity to discuss with Ian the latest thing that I was doing and what I was doing in the work. But of course, it was not a difficult decision to make because, EMBL is definitely one of the premier institutions where anybody would want to go and do work as a PhD student or as a trainee or a postdoc. And as a group leader, definitely. I mean, I would say, yeah, that was the best decision that I took in my scientific career.
Yeah, no, it's a great opportunity to start, of course, a group in such a supporting place. But I was wondering, I mean, as I understand it, you're mostly a biologist, not really a core structural biologist. And were you a bit concerned of coming to a site that was not maybe Heidelberg and maybe had like a very, narrow focus?
Now that's a very good point because my background was not structural biology. It's still not structural biology, but at that time, when I was offered the possibility to come here, I had long chats with the unit coordinator at that time, Stephen Cusack, and we discussed. And I think what I realized is that at EMBL, it doesn't matter in which site you work. I mean, you can consider EMBL as a distributed laboratory space across countries. So essentially I could do everything that I could have done, say, in EMBL Rome or Monterotondo at that time, or EMBL Heidelberg, So that was never a problem because it is the access to facilities across the sites is unrestricted, encouraged, with many different programs that provide collaborative.
And the science community, I guess, is one
exactly.
across the site. But tell me about it. I mean, what were you, imagining to do? I mean, or did you set up shop to do once here? And how did the crystallography or, essentially the structural biology facilities provided you in terms of support to your science?
Yeah. So I think, when I was starting here, so my group was interested, well, my background was interested in studying the role of small non-coding RNAs called microRNAs. So these are found in all cells of a body, and what they do is they select which of the messenger RNAs in our cell has to be controlled and regulated. Essentially, they do a silencing job. So at that time, when I came, there was an area of the small RNAs that was poorly studied, and that's what I started working with. And this was the germline. Why do we have a class of RNAs in the germline that are expressed and they didn't have a clear function at that time? And later on, and now we know that they're involved in genome defense. They help to protect the genome against what we call the mobile genetic elements. So all those things you could do in Heidelberg or in EMBL Rome, but what EMBL Grenoble provided was a good team of groups here investigating structural aspects of different pathways people study, very good biochemistry. So I was very happy to have access to this collaborative environment and be able to use these opportunities. So I definitely try to integrate biochemistry and structural biology into the research that we did. And for that, I should say, we did collaborate with Stephen, we collaborated with Andrew McCarthy here, and these collaborations still exist, even if I'm not here, right?
Yeah, so that's cool.
These are long-lasting.
But one other thing that you mentioned that made me curious, I mean, you talked about the role of microRNAs, and then you said in defense of a germline. So you need a germline. What was the germline that you ended up studying?
Yeah, so that is I should say this is another thing that I think only at EMBL it'll work, because so the model system that I chose to work with is in the mouse germline. Of course, EMBL Grenoble doesn't have any mouse facility, so definitely, the director, the scientific leadership in Grenoble, Stephen, he was very helpful in allowing me access to the facilities around here in Grenoble, but even more, access to the facilities at EMBL Rome. And I have to thank Nadia Rosenthal, who was the unit coordinator there, and in particular, my good friend and colleague, Donald O'Carroll, also an EMBL group leader at that time, for the help to create mouse models at EMBL Rome and then bring them over to Grenoble. So that's what I said. It's a distributed laboratory space, and it never mattered to me that I was not sitting in Rome, where mouse biology is the central focus as a technique, right?
That's great. And I mean, you had even something done in Heidelberg, because I'm a fly biologist, and of course, the fly germline is, of course, I shouldn't say, because not everybody's a fly biologist, but just to say, the germline in the fly is very well studied. So did you look into other types of organisms, not only mouse and vertebrates?
Yeah. So there, I think we had a very fruitful collaboration with Anna Frusi. So there we were looking at a protein called VASA in the mouse germline. But of course, the life cycle of studying mouse is 3 months each time. But then, we realized, actually my student, Jordi Chiol, who was the first author of the study, but that was published finally, he, wanted to work with the Drosophila system. And it was so easy to walk up to Anna Frusi and say, look, I think we would like to do these experiments in your lab. Can Jordi come there? Can you facilitate this? That was no problem. And that is something which still amazes me at EMBL. It's so easy to do.
So maybe, stepping back a little bit more into the biology, one thing that I find very interesting is that, microRNAs are genes in the end, but they make no proteins, right? So how do you see in evolutionary terms these? I mean, were they born even before proteins were existing, and now they acquire this type of role? Or, I mean, do the whole, non-coding RNA biology derives from that, or is a later adaptation? What do you think?
Yeah, so in terms of RNA, I think people believe most of the early life forms were made of RNA molecules, and RNA molecules performed many of the functions that now we know the proteins do very well. So it was an RNA world, and RNA used to be able to do, nuclease functions, polymerase functions. So it was originating initially in RNA molecules. Now, about small RNAs, I think it is a very ancient mode of gene regulation. We can find it in sponges. So it is a mechanism by which cells can identify messenger RNA molecules and then silence them. So it is a universal requirement of any living cell. Most organisms have it, except, let's say, cerevisiae, the budding yeast, that doesn't use the small RNAs. Otherwise, it's
yeah. So maybe this is then in these terms, maybe this is reinventing something that perhaps in bacteria or in single cell organisms was more, geared towards different, or made in a very different way.
Exactly. So these small RNAs that we study in the germline, we call them the PV interacting RNAs, piRNAs. Their role is to identify genomic threats. Now, these are mobile genetic elements called transposons, jumping genes. So all the animals, they have these small RNAs, piRNAs, that identify by sequence complementarity these transposons and silence them. However, even bacteria, they have such threats too. They have transposons. They have invading elements like viruses and plasmids, and they use CRISPR. Sure. So in a way, what we have as piRNAs is the equivalent to the CRISPR system. And now, after all the research that we and the entire field has started in 2006, it's clear that if you look at how these piRNAs are made and how the CRISPR RNAs are made and function, the logic is same. Everything is same, just that how it is done is different.
Sure. So it might have been evolved, different times with different starting material, and it is fascinating because it's basically telling you that evolution tries to sort of answer to a need that the organism had, and in this case, probably this need came up at different times during evolution. Protecting your genetic information is, I guess, very important for life.
So the need was very simple. How do you specifically identify a threat? So you need a sequence, you need an RNA molecule, and this is then hooked up with a protein that is the executioner. It's a nuclease, either cuts DNA as in CRISPR or RNA as in the PV interacting RNA pathway. So in both cases, the cell is searching for solutions. You have these various different kinds of transposable elements with different sequences. You rely on sequence.
Yeah. What I found interesting in these systems is that it almost, this is general for very complicated evolutionary solution, I guess, that it seems that you have almost an intelligence there, right? You have a problem and there are some solutions that are pretty complicated, right? Talking about this, for instance, how do you distinguish from, a foreigner DNA from an intrinsic part of your genome? How do you distinguish between a DNA part from an RNA part? These, the evolution has found solutions, but they are not trivial solutions, right?
Yeah, no, so I think that is something that we found here at EMBL, like the piRNA pathway actually has what one could consider as an adaptive immunity, So the germ cells know who are the existing threat, whatever is in the genome, and it has developed mechanisms to target that, and we could call that the innate immune system. So it knows what is there, it has handled it. But it also has this adaptive arm of this immune system where we found that when a new transposable element or new sequence comes in, it can identify this new threat and produce small RNAs against that threat. So, I'm not going to go into details of how it's done, but it shows that it has the versatility to adapt and then source information from the threat itself and then fight against it.
Use almost the biological material because in origin it has to, chop that probably piece of invading nucleic acids.
To make new small RNAs. Yeah. This is the amazing aspect. You asked me about the comparison to prokaryotes and eukaryotes. CRISPR does exactly the same. Every time a bacteriophage infects the bacteria, it takes a little fragment of the phage DNA and keeps it as a memory in its genome, in the CRISPR arrays. And then it uses that as a mechanism to fight back future infections. Our cells do the same, but it uses not CRISPR, piRNAs. So, and they have evolved different ways. The sole purpose is still the same, right?
So, of course, you're interested, I guess, in the fundamental science of this, but as in the CRISPR example, are you thinking also in terms of technological or medical use of these parts of biology?
So that is something, when we made the studies here in Grenoble, so essentially what we showed is we can take any piece of DNA, let's say luciferase or GFP, and make piRNAs from it, small RNAs that could function. So we call it artificial or designer piRNAs, which means you can now use that to target any gene of interest and then have silencing effect. But that is the theory, and you could show that it works to an extent, but to apply it is difficult because it's not that simple as the CRISPR-Cas system where you have a protein and one RNA, that you need. You need a whole machinery of proteins to do it. So yes, I think we thought about using it as a technology for silencing genes. We even tried to see how we can, if it can't be done easily, can we make it work in a system? But still, it's not possible. Even after so much. It's complicated because you need like almost 25 proteins.
Okay, so that
to make those RNAs.
From a technological point of view is quite an elaborated system. But you said yourself you use the mouse as a model system. So I guess the idea is that there's probably some connection to humans or to disease in humans. So is it something you're looking into?
Yes. So I can say that almost every gene that we identified and studied to be involved in this germline small RNA pathway, people have found mutations in humans that lead to infertility. So I didn't mention this, that these germline small RNAs, if they don't work properly, then the germ cells die. They're not made because of genome damage. Because the transposons are hopping around, they cause mutations and the germ cells die. And which means there's no sperm, there's no egg. Right? So people have identified mutations in every single gene that we and others in the field have worked on to be involved in human infertility.
So if you think back at your time at EMBL, I mean, what do you think is, was there a aha moment in your research or something that you remember in those 10 years as very, very prominent in your science?
Yes, so I would say, of course, like many other groups studying in the field, we were trying to understand, how these germline small RNAs are made. I mean, we knew quite a lot about microRNAs, which are found in every cell of a body. But it was in 2006 that people started to realize that we have a class of RNAs in germ cells that are totally different. Their purpose is to protect the genome. So yes, we and others found different factors that are involved in the production of these RNAs. But I would say one thing that I remember, which really was an exciting finding for us and the field, would be that the fact that a new target that is coming into the cell, once it is attacked by these germline small RNAs, that target itself becomes a source of new piRNAs that are made.
So the adaptative part.
Adaptive immunity. So this was like a way that, what does the germ cell do if it has never met a threat? What if it is faced with a new threat, and how does it handle it? Well, the answer was simple. It takes a trophy. It essentially chops up the fragment that the RNA that is coming in, and it takes little pieces of it as a memory. So it stores this information as a memory and then uses that for further silencing.
It's almost a form of learning.
It's a form of learning, yeah. So that is kind of the immediate short-term memory, and the long-term memory comes when these elements that come in, they insert themselves into the genome, because that's where you have to record the memory, right? Like in the bacterial system, the CRISPR, bacteriophage pieces have to be stored and then used.
So the catalog of threads.
It's actually like a library. It's a library of fragments of thread that is coming in. Each book is one thread.
Yeah, it's amazing how, this is not, only biology, but I guess it's mostly biology. But it's amazing how, things have maybe similar meanings, but the meaning is completely independent of the medium. I mean, we are talking about learning, we are talking about experiences, but we are talking at a molecule of DNA, but we could be talking about, our brain. It's amazing how there is a sort of, concepts that are transcending, the means of biology, don't you think?
No, I mean, in terms of the basic concepts are always the same, right? I mean, how this information that is obtained by a particular cell type, that information is passed on. For example, in the case of the germ cells, we can say that we inherit the silencing that was established in the germ cells in our parents. That silencing information we inherit, and we pass on the information that we put in, in the germline genome using our germline small RNAs to the next generation.
Yeah, some people would say that this is another form of epigenetics.
It is epigenetics. It's just that the small RNA is like the postal code, pin code. It tells where to silence in the genome and what marks to put in there, and that is faithfully transmitted to the next generation.
So is it going only one generation, or it's like a additive, progressive system that takes into account all that has been learned before?
Yeah, so that's an interesting point. So we know that the information that is inherited from the parents, we acquire, but our germ cells, they go through a process of rebooting, and we call it reprogramming, germline genome, epigenome reprogramming, where all these marks are erased and they reset again, right? So in each germline it's reset. So yes, it goes to one generation and then you reset it because who knows, there's a new threat. So you got to incorporate that into the new genome that is made and transmitted further.
That's cool. Yeah. So how do you find now being in a university, the environment different compared to a small EMBL site? I mean, what are the differences, the pros and cons?
So in terms of the major difference is teaching. So university expects you to be teachers, professors, giving lectures and classes. So that is the primary job that we are employed for. So that is a major thing, but I should say that I love it.
So I mean, I'm curious because I also do, being in a university now, a lot of teaching, and I also enjoy it. But one thing that I realize is that it's somewhat helping my science. Is it? I mean, the fact that you have to explain yourself to students that have, of course, a much lower scientific background at that time. Is it something that helped your science in thinking what would be the experiments to do and the question to go after?
Yeah, absolutely. I mean, students, so I teach at the bachelor level, third year, and, you better be ready. Sometimes the questions are so fundamental, and you think, oh, well, yeah, that's true. I mean, I never thought of that, right? So, I mean, because we dig so much deeper into our problems, and then we focused on, what one would consider details, but, teaching students always brings us back to the basics, and then that always helps, yeah.
Good. So, another standard question that we always ask is what EMBLs mean to you.
So I would say, me personally, giving opportunity to a postdoc to start a research group and give all the means that are required to facilitate that. Yes, there's the financial aspect, the support that EMBL provides, the infrastructure. I mean, without the amazing core facilities that EMBL provides, both in Heidelberg and other places, in Rome, as mentioned already, the mouse facility, structural biology environment here, it would be difficult to do. So, but more than that, I think the people at EMBL, the scientists at EMBL, and I always tell my students and postdocs in the lab in Geneva, if you're looking for a job, go and try to get a position at EMBL. Definitely a good place to do it.